Hi, welcome to another episode of Catch This. My name is Trudy Ray, and today I would like to talk about a story that is making the rounds linking SARS-CoV-2 vaccination and infertility. The claim is that there is a certain level of sequence similarity between the SARS-CoV-2 spike protein and syncytin-1, a protein required for placental development. And the concern is that if immunization with a vaccine induces antibodies to the SARS-CoV-2 spike protein, then those same antibodies would also attack syncytin-1 and thereby prevent formation of a placenta. It is reasonable and even advisable to question the safety of any new vaccine. However, in this video, I want to walk you through the facts and explain why these concerns regarding syncytin-1 are unfounded. Let's start with some background information about syncytin-1. This is a protein that mediates cell-to-cell fusion, and in the context of pregnancy, it mediates the fusion of trophoblasts, the cells that form the outer layer of a blastocyst. A blastocyst has an inner cell mass that ultimately forms the embryo. During that development, the trophoblasts provide nutrients to the embryo and develop into a large part of the placenta. So there is a claim that there is a certain level of sequence similarity between this protein, syncytin-1, and the SARS-CoV-2 spike protein. Where did this claim originate? I'm not sure if the whole thing started with this blog post, but I think it had a lot to do with it. It was a post on a website called Health and Money News and was titled, "Head of Pfizer Research, COVID Vaccine is Female Sterilization." Whoa! First of all, that looks like clickbait to me, but I am of the opinion that extraordinary claims require extraordinary evidence. So let's do the work and at least attempt to find the evidence for this claim. First of all, who is this head of Pfizer guy? It turns out to be Mike Yeadon, who, according to his LinkedIn profile, used to be the Chief Scientific Officer of Allergy and Respiratory Research at Pfizer 10 years ago. But he's no longer there. So now, in this blog post, he's allegedly making the claim that the vaccine contains a spike protein called syncytin-1, vital for the formation of human placenta in women. If the vaccine works so that we form an immune response against this spike protein, we are also training the female body to attack syncytin-1, which could lead to infertility in women of an unspecified duration. For starters, there is already a lot wrong with that first sentence because although the vaccines do contain DNA or RNA that codes for the SARS-CoV-2 spike protein, the spike protein is not called syncytin-1. The vaccine does not contain syncytin-1. There are no placental proteins or genetic material that instructs the manufacturer of placental proteins in any of the vaccines for SARS-CoV-2. For a review of what the mRNA vaccines do contain, I recommend my previous video linked here. So at this point, I'm already highly suspicious and I start to question the legitimacy of this website. What is this news source, Health and Money News? It doesn't take too much work to figure out that it is a website generally devoted to conspiracy theories and misinformation. All right, moving on. Let's take a look at these sequences to see if there is indeed a similarity between the SARS-CoV-2 spike protein and syncytin-1. How do we find this information? There's a website called GeneBank. This is an open access annotated collection of all publicly available nucleotide sequences and their protein translations. It is produced and maintained by the National Center for Biotechnology Information, NCBI, which is a part of the National Institutes of Health in the United States. This is funded by your taxpayer money, and everybody has access to this website. Everything I'm about to show you, you can do yourself and verify for yourself. If you type in SARS-CoV-2 spike protein, it will bring up the 1,273 long amino acid sequence. We know this is a protein sequence, not a DNA or RNA sequence, because we see letters like Q and R in there, for example. If it was DNA or RNA, there would be only A's, G's, C's, T's, and U's. It's the protein that we want, so this is good. Now to find the amino acid sequence of human syncytin-1. There it is. It is 538 amino acids in length. So now we can do an alignment of these two sequences to see how similar they are. There are multiple websites that you can use to do that. One common one is BLAST, which stands for Basic Local Alignment Search Tool and is also operated by NCBI. To align two protein sequences, we have to do a protein-protein blast. So you copy the SARS-CoV-2 spike protein sequence from here and paste it into this box right here. And then you take the syncytin 1 protein sequence from here and paste it into the second box here, and then click blast. And we find that there is no significant similarity found. The thing is, if there are very small sequence similarities, they will not be detected by the BLAST algorithm. So we already know that this algorithm does not consider these two sequences to be particularly similar. However, let's take it a step further. There are other algorithms that can be used. Here's another one I like to use called EXPASI, which stands for Expert Protein Analysis System, and is operated by the Swiss Institute of Bioinformatics. This is a great resource for achieving many different things within the realm of bioinformatics. But again, we just want to compare two protein sequences. So we are going to copy the spike sequence and paste it in here. Then we will copy the syncytin sequence and paste it in here. And then we click submit. And then what you get is a bunch of different alignments where the computer found a certain % similarity between a portion of the spike sequence and a portion of the syncytin sequence. Each asterisk denotes a match between the two sequences, where the amino acid is the same. The first one here has the highest score, which means that it has the biggest number of matches in the two sequences. And this is the one that Yaden focused on in a December 2nd tweet. And I'm not quite sure why. It's a very short sequence and has no particular immunological relevance over other sequences. It's not part of any specific epitope or portion of any antigen that is recognized by antibodies, B cells, or T cells of the immune system. And just to illustrate how irrelevant these similarities are, I'd like to walk you through a string of tweets by Swiss immunologist Andrew Croxford. I think he did a fantastic job illustrating his point. He selected several proteins that are very common in our cells and did a similar alignment with the SARS-CoV-2 spike protein. There's actin, a very abundant protein in human cells that is found in muscle cells, for example, and we see regions of similarity that are similar in size to the one found between spike protein and syncytin. Or there's collagen, a very common structural protein found in connective tissue. And again, there are 4 amino acids aligning with a gap in between. Or hemoglobin, an iron-containing oxygen transport protein in our red blood cells. Again, we see a similar pattern. If the vaccine elicits antibodies against syncytin 1, then it would make sense that it would also induce antibodies against these proteins as well. Here's a great analogy by Breon Barker on episode 703 of This Week in Virology.
I haven't looked at the data on syncytin in a while, but I think when I last looked at it, the similarity that was discussed here was a sequence of 4 amino acids in a row between syncytin 1 and the spike protein. And the way that I described that to a non-science friend was noting that it is probably likely that if you looked through all of the things I've written in my life and all of the things that Shakespeare wrote in his life, you would find a place where we use the same 4 words in a row. But that does not mean I am in any way, shape, or form Shakespeare. And this is not going to be enough of an overlap or enough similarity in amino acids to really lead to an antibody response that would be cross-reactive.
Shakespeare. That was brilliant. There is simply not enough similarity in amino acids between the SARS-CoV-2 spike protein and human syncytin 1 to elicit an antibody response that is cross-reactive. Furthermore, while T cells do recognize linear epitopes, meaning that you can just use the sequence to see if there's significant similarity, B cells recognize native proteins, which are folded. And this folding may put amino acids next to each other that are far apart on the actual sequence. And although most B cells do require help from T cells to respond to antigens, and while there is a linked recognition that ensures that B cells and T cells respond to the same antigens, these regions of similarity are still very small, much smaller than the typical size of an immunogenic epitope. One other point. What about the thousands of women in the past year who had COVID-19 before or during pregnancy and then delivered healthy babies? According to the CDC COVID data tracker, over the past year, there have been over 53,000 COVID-19 cases among pregnant women. If anti-SARS-CoV-2 spike protein antibodies produced by the mother were attacking syncytin 1 and thereby the placenta, wouldn't we already have seen an abundance of pregnancy complications and miscarriages? So far, there has been no evidence of that kind reported among pregnant women. So why focus on this particular protein in making a claim that any SARS-CoV-2 vaccine might be potentially harmful? I have my suspicions. The assertion that any vaccine causes infertility in women is not new. This has happened all throughout history and all over the world. For example, there was a conspiracy theory promoted by Catholic bishops in Kenya that the tetanus vaccine is for racist population control, and also the false claim in Africa and the Philippines, among other places, that the polio vaccine can impair female fertility. The polio eradication campaign in some Muslim countries has long been hampered by conspiracy theories that claim that the polio vaccine is laced with anti-fertility chemicals that can render young girls sterile before they even become women, and or that it can also infect them with HIV as part of a bigger plot to depopulate the developing world. But perhaps the vaccine that is most commonly falsely cited by members of the anti-vaccine movement as causing female infertility is the HPV vaccine, particularly Gardasil, which has been blamed for causing premature ovarian failure without evidence. And apparently that is also a population control measure. So it's really no surprise that this claim has resurfaced in the context of this new vaccine. Unfortunately, the anti-vaccine movement is a well-funded and well-organized lobby with hundreds of websites that are backed by political action committees, social media, and poorly informed celebrities with loud voices. And sometimes they try really hard to sound science-y. However, this particular claim does not hold water. Long story short, the SARS-CoV-2 vaccine does not contain syncytin 1. The SARS-CoV-2 spike protein is not syncytin 1. And there is insufficient similarity between the SARS-CoV-2 spike protein and syncytin 1 to warrant any concerns that any antibodies induced by the spike protein vaccine would attack syncytin 1, thereby leading to termination of pregnancy. And please keep in mind that the human body has systems in place that stop immune reactions that might harm its own tissues. If we didn't, we wouldn't even make it past day 1 in life. That's it for today. My name is Trudy Ray. I'll see you next time on Catch This.